Maximo Petarin is a PhD student at Goethe University, Frankfurt, and he is going to be presenting today on a combined in vitro in silico approach to predict the oral bioavailability of borderline BCS class 2, class 4 weak base albendazole and its main metabolite, albendazole sulfoxide. The virtual floor is yours, Maximo.
So thank you very much for the kind introduction, John. My name is Maximo Petarin. I'm a PhD student at the Goethe University in Frankfurt am Main, and today I will be presented a project supervisor, Dr. Edmund Kostovitz, and Michael Bolcher from Simulations Plus regarding the prediction of the in vivo performance of BCS class 2 borderline 4 drugs using a combined in vitro in silico approach, with particular focus in albendazole and albendazole sulfoxides, which were the study compounds. So if we can go to the next slide. Thank you very much. So the general objective of my work was to use a combined in vitro and silico approach to accurately predict the plasma concentrations of albendazole and its main metabolite albendazole sulfoxide after the oral administration of a tablet formulation in the fasted state. If we can move to the next slide. Thank you very much. But before we go into the details of the PBPK model, it's important to describe the compound of study itself and to describe any kind of considerations that we have to take into account while setting up the PBPK model. So to start with, we can say that albendazole is a BC4 drug. This classification is borderline due to the lack of conclusive permeability in humans. Albendazole has an acidic pK of 10.26 and a basic pK of 4, meaning that this molecule is actually an ampholyte, but in vivo behaves as a weak base, because in vivo we don't find any pH values at around the pH range given by the acidic pKa. So therefore, the basic pKa is the one that counts. Albendazole is a highly lipophilic drug with a relatively high log P of around 3.56, and therefore exhibits an extremely low intrinsic solubility of around 1 microgram per ml. Not only this, but being a weak base or behaving as a weak base, albendazole solubility decreases with the increasing pH within the gastrointestinal tract when moving from the stomach into the intestine, and therefore we will expect supersaturation and precipitation upon gastric emptying. On top, albendazole experiences an extensive first-pass metabolism close to 100% by different enzymatic systems to its main metabolite albendazole sulfoxide, which is the only metabolite that is claimed to be detected in plasma and to which therapeutic activity is attributed. Therefore, we also have limited information in the clinical studies. To start with, we have a lack of intravenous data from both compounds. This means that we have to work with predicted volume of distribution values that we predicted using GastroPlus. We only have one clinical study in the literature that published both albendazole and albendazole sulfoxide plasma concentration profiles, and the other clinical trials that we found in the literature, in which only the metabolite concentrations were published, were used for further model verification. All in all, we need to say that when setting our PBPK model, the pre-absorptive scenario, that is the absorption, was governed by the physicochemical and biopharmaceutical properties of the parent compound, while the post-absorbed scenario, this would be correct, yeah, the post-absorbed scenario was determined by the PK parameters of the metabolite albendazole sulfoxide. If we can move to the next slide, please. Thank you very much. Therefore, to go into the key inputs of our PBPK models, we first have the determination of albendazole's solubility, which we did by determining the thermodynamic solubility of albendazole in biorelevant media. First, what we did is we determined the solubility in biorelevant buffers, as we see in the table that we see in the left-hand side, in which we determine albendazole solubility in a series of buffers that have the same composition as the intraluminal fluids that we found in the gastrointestinal tract in terms of pH, buffer capacity, and also ionic strength or osmolarity. And as we can see in this table, we see that albendazole solubility is high within the gastric pH range from a pH of 1.3 to a pH of 2.0. But the interesting part is that albendazole solubility drastically drops even within the gastric compartment, within the pH ranges that we usually found in healthy volunteers in the stomach. And its solubility further drops once we move into the intestine to 0.53 micrograms per ml, which is extremely low. So with these values here that we determine in in biorelevant buffers, we design our pH solubility curve, and we observe that albendazole eventually actually presents a high dependent pH solubility. And from these values, we also determine our pKa in an experimental way that we were able to fit into GastroPlus to have an accurate model to predict solubility as we run our simulations. Furthermore, given that albendazole is a highly lipophilic weak base, it is normal to expect that there will be an extra solubilization effect given by the bile salts present in the intestine, and therefore we carried out more solubility tests in biorelevant media containing bile salts, as we can see here in the right hand side. This FASIF media in the upper square and FASIF media and SGF are all biorelevant media that intend to simulate the composition of the intraluminal fluids in the stomach and the intestine, which in this case contained bile salts. And we see a solubilization effect by means of these bile salts in the media. And this solubilization ratio could be quantified, precisely quantified with GastroPlus, and used as an input in the model. If we can go into the next slide. Thank you. Therefore, given that albendazole solubility depends on the pH that we have in the gastrointestinal tract, we decided to carry out transfer model tests. For those that are not familiar with the transfer model setup is a two-stage dissolution setup initially introduced and developed by Dr. Kostovitz that uses the current USB 2 apparatus, in which we have a donor phase, and as we can see in the figure, that contains gastric media, where we introduce our formulation. It could be a tablet formulation, capsule, powder, granules, etc., any kind of oral formulation. And when the formulation is allowed to disintegrate and dissolve, it is simultaneously transferred by means of a peristaltic pump into an intestinal compartment or an acceptor compartment that has intestinal media. The transfer is done by means of this pump, which is programmed to have a biorelevant or physiological gastric emptying rate. Therefore, from this intestinal compartment, we take samples, we analyze the dissolved concentration, and we build transfer model plots or transfer model profiles that we see in the left-hand side. We did this with albendazole at two different dose levels, which are 400 milligrams in dark blue and 800 milligrams in light blue, and which are the doses that we have in the clinical trials that we found in the literature. And what we can see here is that albendazole clearly supersaturates upon gastric emptying, clearly supersaturates over the thermodynamic solubility that we see here identified with a dotted line, and precipitates immediately following a first order precipitation rate constant that we were able to quantify and also include into GastroPlus. GastroPlus offers the possibility to include precipitation kinetics into the models to make them more accurate for this kind of compounds. So if we can move to the next slide. Thank you. So considering these two main key parameters as inputs into GastroPlus, we were able to carry out our initial simulations, our final simulations with our final PBPK model setup. And what we see here in the left-hand side are the simulated versus observed plasma concentrations of albendazole, that is the parent compound. And on the right-hand side, we see the metabolite albendazole sulfoxide. And what we can see that in both cases, we were able to accurately predict both plasma concentration profiles. It is also clearly observable here, one can observe clearly that with the parent compound, we were unable to perfectly fit the plasma, but we need to take into account the huge inter-individual variability that we find with this kind of compounds, given the erratic dissolution to which they're subjected, due to, as I said before, supersaturation and precipitation or variability in the pH values within the gastrointestinal tract from a healthy volunteer. So even though we were able to actually predict the plasma concentrations within the given range, we are within the inter-individual variability, and we were also able to correctly capture the metabolism of albendazole into albendazole sulfoxide, as we can see again in the profiles. But as I said before, we have to use predicted values for volume of distribution, and therefore, given that we use a predicted value that was not experimental, we needed to challenge our PBPK setup with GastroPlus against different sets of clinical data, and this is what we can observe in the next slide. Thank you. I know. We can move forward one slide. And one more, please. Thank you very much. And here we can see how we challenge our PBPK setup against different sets of clinical data, 3 different sets in this case, at the 400 milligram dose level, where we see the concentrations of albendazole sulfoxide, which, as I said before, were the only concentrations that were published. And if we move one more slide, we also see that we challenge the same PBK setup with GastroPlus against clinical data sets, in this case at the 800 milligram dose level. And within the simulations result, we were able to confirm that our model can accurately predict the plasma concentration profiles of both compounds, especially of the sulfoxide. Again, given the complex scenario represented by these compounds in the absorption and post-absorptive phases, and also given the high individual variability. So if we can move into the final slide, I would like to conclude this work by saying that we were able to develop a accurate and solid PBK model with GastroPlus using a combined in vitro and in silico approach, for which it was also crucial to consider and to include supersaturation and precipitation kinetics into this model, that we were allowed to do it by using GastroPlus. We also did a critical assessment of different parameters, of other input parameters, such as the metabolism and permeability, that in this case I couldn't include due to time restrictions. But the solid and the validity of the in silico setup was also further verified by using different sets of clinical data. In this case, we used as many sets of clinical data as we could find in the literature, despite the lack of IV data and PK parameters for these compounds. So I would like to conclude my presentation by, we can go to the next slide, by saying thank you very much for your attention. And if there are questions, I will be glad to answer them.
Okay, Maximo, thank you very much. Similar to Al previously, very efficient use of time and presenting a great deal of information.
Thank you very much. I apologize for the technical inconveniences.
No worries.
So,
not sure if, well, actually, one question while I get our next speaker set up. Did you, knowing who was assisting you on some of these activities with the GastroPlus modeling, our founding scientist, Dr. Mike Bolger, were there efforts to explore the use of the mechanistic nucleation approaches for precipitation kinetics with the data that was generated?
Yeah, this, John, is a very good question. This is our follow-up work. We are using the current mechanistic and nucleation growth model equations, for which we need much more experimental data, but we're in the way, because as probably some of you know, if you're able to determine the parameters that fit this mechanistic and nucleation growth model equations, you will be able to set them for one dose and for other dose levels as well, without the need of repeating this kind of in vitro experiments that I did. So for example, if we can set up these parameters for the 400 milligram dose levels, and because of some of the parameters, of course, are constants, we will be able to apply this into every kind of dose level, or even within any kind of same type formulation to get good predictions. So the question is, yes, we're considering this, and this is the follow-up work to this project.
Excellent. Look forward to seeing it presented at MIDD Plus 2022.
Yeah, certainly.
Very good. Well, thank you again, Maximo. We appreciate the time.